recording. So this will go here. Yeah, absolutely.
People will get candy if you get questions right.
Or if you answer.
Or if you
just participate.
Too bad. Take it. Yeah. Basically. But yeah.
Can you throw it like at like right here? Hopefully it'll take me out so much.
If you participate.
I tried.
You got this.
Where are these slides gonna be on the announcements?
I'll send them to the announcements, yeah. I'll also put a recording
That's your phone.
link or something. I'll figure out how to do that, yeah.
Yeah. We upload them to the YouTube, and then
Oh,
we
is
can
that
link
how
the
we do
YouTube.
Okay.
Yeah.
Good call. I was gonna say, like, I don't want to download a file.
If you also want to like look at the old review sessions, old Echo 360s. Low-key old Echo 360s save me. I love them so much. They're so funny. And I just like see myself spinning in my chair. That always happened to me in this class too. I like you know the chairs in Morrow, how they like spin. I was like always like moving back and forth like the whole class. I was the only person moving too.
I hate that classroom though.
I hate it. I hate it so much. Alright.
Yeah.
You want to start?
Yeah. All right, guys. Thanks for coming. This is topics 1 through 5 review. Tomorrow, me and Niall will be, I think, here again.
Yeah.
Doing 6 through 10, so please come by. I have a lot of candy, so please participate. Ask any questions. I'll huck some Three Musketeers at you.
And if you have any allergies, just let us know. We can give you
Yeah.
candy that's not gonna cause you allergies.
All right.
Yeah.
Any questions before we start? Anybody have any like burning exam-related questions? All right.
Cool.
Cool.
Yeah. Okay, so we made this slide for general goals and advice, just like study tips. I would say the first advice is to understand the basics first. So if you're confused about like the general ideas behind a topic, don't worry too much about like the really specific examples that like you might see. Because if you don't really understand the basics, they're gonna confuse you more. Start with the basics and don't stress too much, because if you know the basics, you can apply those concepts to the other things also. Ask your friends or TAs for help. I know a lot of people love ChatGPT. You all are like, yay, ChatGPT, explain it to me. But sometimes ChatGPT is wrong. And I love the videos that are like, ChatGPT, tell me something. And then they copy the answer ChatGPT tells you, and then they paste it and send it back. And they're like, is this right? And then ChatGPT goes, no, it's not right. It's like, don't trust it. Unless you can fact check it yourself, you don't really have a way of knowing whether it's telling you true information. But if you're already confused, you can't fact check it, obviously. So my personal opinion is use your resources, use your friends who know what's going on, ask the TAs, email us, come to office hours, ask Laura, email her. She likes responding to emails. She will respond to you. But, you know, use trustworthy sources, and that's the second one. You can also just Google things. Google's pretty trustworthy. You can just look for things, use trustworthy sources on Google. A lot of images help too.
Yeah, rewatch lectures and the pre-lecture videos, because that's how I pass this class. Those are like, if you can rewatch them and understand everything they say, then you're in like a golden spot.
Yeah. Rewatching lectures is always really helpful. Try not to overcomplicate it. Like I said at first, don't get bogged down in like the details first. Look at the general pictures and try to understand them, and talk about it a lot. Practice active recall. Like discuss it, chat about it with your friends. Try just, actively remembering and talking about it without looking at your notes too much. It'll help.
Yeah, cool.
Yeah.
All right, so we'll get into topic 1 first. It was just the, go for
I
it.
just have one question. How crazy are these multiple choice questions going to be? Like, are they going to be like 8 answer choices with like 2 or 3 that are going to be correct, like the quick questions in class?
No. I don't think Laura's a big fan of those tests.
Yeah, it's gonna be a lot of, it's not gonna be like memorize, this pathway. It's gonna be like, okay, you have a situation, what could be the possible result?
Yeah.
Okay.
That
Think
sort of
about
thing.
Multiple choice questions you might have seen. I know she's kind of backed off of the multiple choice a lot in her classroom setting, but the final is just really hard to do a whole big exam and grade it, so we do a lot of multiple choice. But I would say trying to focus on where the question is coming from first, and then getting into like what you think the answer might be is helpful, but it's not going to be like super overwhelming, I don't think.
Okay.
Yeah, they shouldn't be super scary. Nothing like the, is this yes or no, and then it's maybe. Nothing like that.
Yeah.
Thank you.
Yeah, do you want me to post it now or after?
I think people like beforehand. Yeah.
Yeah.
I can post
Do you
it.
wanna
I
do
can
that?
post
Yeah.
it. You want me to talk about topic 1?
Yeah, just make sure when you download it, I know we didn't finish the 6 through 10 yet, so just delete those. before
Got
you
it.
put
Got
in,
it.
okay?
I'll only download.
All right, we'll put that up shortly.
Yes.
Yeah.
Working on it.
Anybody else? All right, sweet. All right, topic 1. It was a while ago, but you need to know the basics for the exam. So run through it yourself after the review session too. Just make sure you have this down. This will lay the foundation. This and the next couple topics will lay the foundation for the rest of them, as you guys know. We've put these unit objectives on every slide, and once they get published, you guys will see them. But a good way to study is to go through all the unit objectives and make sure you're comfortable with explaining each one of them. And then you'll be pretty much set for the exam. So I won't go through all of them right now, but you guys can look at those on your own time. Quickly, we'll go over prokaryotes and eukaryotes. These are the main differences and similarities that you guys should you should have down pat for this exam, like single cell versus multi-celled. Who can tell me which one is which? You'll get candy.
Prokaryotes are single celled.
Yep.
Yep.
My bad. And yeah, you guys can figure out the rest of the list, but remember that no nucleus or organelles in prokaryotes. They're just kind of like a blob, a single cell with everything housed inside of it. The big thing is transcription and translation happens concurrently. I'll get into that later in the slideshow, but that's one of the big differences. And then in eukaryotes or multicelled, they have membrane-bound organelles and a nucleus. That's the big thing that differentiates it from a prokaryote. And then the opposite of the transcription and translation, those happen separately. But Laura might have a trick question in there about the similarities, so you should know that both have DNA genomes. Both carry out gene expressions, and ultimately that results in them making proteins. Those are consistent across all cells. Make sure you know that. This can confuse a lot of people. They both have a phospholipid bilayer surrounding the whole thing. They're slightly different in each of them. You don't need to know the differences, but you should know that they have a phospholipid bilayer. And then you might see this somewhere in your Googling or Khan Academy, but some types of eukaryotes and some types of prokaryotes have cell walls. It's kind of like a spectrum, so you don't need to really know that, but if you see that, don't get confused by it. So yeah.
All right, slides are up
Okay. Yeah.
on Canvas under announcements if everyone.
All right, can somebody tell me why prokaryotes do transcription and translation at the same time, but eukaryotes cannot?
Because prokaryotes don't have membrane-bound organelles, they're able to do transcription and translation both in the cytosol.
Mm-hmm. And
Whereas eukaryotes have membrane-bound organelles, so that they will do transcription between the nucleus, and they have to go through RNA processing, and then translation in the cytosol.
perfect. Good job. Yep, just like she said, they happen in separate space and time in eukaryotes, and prokaryotes do it all together. That's the main thing. You got a good explanation, so good job.
In the back, do you have a question?
Do eukaryotes also have to do different spots for transcription and translation because they're more complex and that's not?
Because they're more complex, they keep their DNA more organized inside of the nucleus, so to protect it, because it's a lot larger, which is what's physically stopping them from doing transcription and translation in the same place, because the ribosomes aren't in the nucleus. Does that help?
Okay. All right, so this is a quick diagram. You can look at this again in the slides, but these are all the pretty much main differences between a prokaryotic and eukaryotic cell. If you go through it, kind of you can see the different organelles and stuff. Just have a general idea of what most of them do. Again, you're not going to be quizzed on what does the Golgi do, but you're going to have to know that the prokaryotes don't have a Golgi necessarily. You'll also need to know the differences between cells and viruses. So viruses are cell-specific obligate parasites, which basically just means they require a host to live and duplicate. So they're not considered cells technically, even though they have kind of a layer and stuff in the middle. They're not considered cells because they're not considered living. It's a big question in science, but for this class purposes, they're not considered living, nor are they considered cells. They're made up of a protein coat and sometimes have like a membrane outer layer, but sometimes they don't. They have some type of nucleic acid genome. So some of them could be double-stranded DNA, some of them could be single-stranded RNA, double-stranded RNA, which is really uncommon, but it's possible. They have something to carry genetic information. The three main things you might see for like a short answer question or maybe even a multiple choice: they don't grow, they don't carry out metabolism, they need a host cell to do that, and they don't have an energy source, which are three reasons why they're not living. So make sure you know those three. Any questions so far?
Is the only reason that there can be considered living is that they can interact with their environment?
So technically they interact with their environment already, but they don't grow on their own. They're not self-sustained.
I think, yeah, sorry.
When we go through the topic, it talks about like why they would be considered living and why they would be. The non-living would be because they need a host for pretty much everything, except they can by themselves interact with the environment.
Yeah. So, yeah, well, it might seem like they have qualities of certain living organisms and certain organisms that are also considered living. If you look it up, there are actually a lot of overlaps between other organisms that are living, some organisms that do require a host to replicate, but are also living. But there are a lot of counter arguments as to why they could be considered like a living species, even though they're technically not really considered living. So if you get a question on the exam about it, you could just make arguments for both cases and then say, but because they're mostly non-functional on their own, they're not living.
But I could say because they interact with their environment, they can be life.
Yeah.
One reason why they would be considered living.
Yeah, that you could argue that they resemble living organisms because of their interaction with their environment. Yeah, for sure.
Did you have a question as well? Did anybody else have a question? Did you have a question? Okay, cool. So gene expression is kind of like the main thing that you guys have studied in various contexts, but it was introduced in topic 1, so I'll go over the basics. Information that's stored in DNA, in the DNA sequence, becomes RNA, which then becomes amino acids, which then can become a protein. Proteins carry out various functions, but the information is housed in DNA. The main thing you should remember is just transcription, translation, protein. That's the main thing. Like, that's the way it all works, kind of. Yeah, transcription, as you guys know, it's DNA to RNA. Translation, RNA to an amino acid sequence, and then the protein's made when that amino acid sequence folds. And everybody can shout this out. Protein folding determines what? Function and structure, yeah. Protein structure. And then the last thing we talked about, I think this is the last thing we talked about in topic 1, stem cells. You guys should know the different levels of potency and kind of what that means for what kind of cells they can become in the future. So the main three are totipotent, pluripotent, and multipotent stem cells. These cells are found anywhere, but again, the totipotent and pluripotent cells are only found in developmental stage. Multipotent are the only cells that exist in adult bodies. Yeah, descriptions are here of what each of them do. The totipotent cells have the potential to differentiate into any type of cell because of how early they exist in development. Pluripotent cells can do the exact same thing except become extra embryonic tissue. And then multipotent stem cells can become a less kind of variety of different cells, but they exist in adult cells. It's kind of the only thing you need to know. You might get a question on where they're found, which will be anywhere that can regrow new cells at a high rate. So your hair, your skin,
Liver,
bone
blood
marrow,
cells.
even your tongue. But yeah, those are the main 4.
Because having potency is the ability to replicate.
Exactly. Cool. Any questions so far?
For the 2 defining characteristics of stem cells, we would say ability to self-renew or
Mhm.
replicate and ability to differentiate.
Correct. Cool.
Any overall topic 1 questions now before we move on to topic 2?
Will differentiated cells be on?
Sorry?
Differentiated cells, will that be mentioned in the topic 2? I know it was brief about differentiated cells, but will this be about stem cells?
So, I mean, stem cells are just they're unique in the fact that they can differentiate. Any other cell type, you can consider them differentiated. So, like as a general theme, you can think about it as like any normal cells you talk about have already been differentiated. Anyone else?
For the virus, is it okay if you say that they live like in a space between like living and non-living? Is that
Yeah,
totally fine?
that's
Sure.
that's perfectly valid as an argument. For the virus topic, for discussion, if you're talking about it, you just need to be able to justify your points and talk about it with like with a grounded point. You don't need to like say there's no one specific answer we're looking for. You just need to be able to like talk about it knowledgeably.
Okay, thank you.
Okay. Topic 2. Amino acids and protein folding. So here are the unit objectives. You can look at them. And then these are helpful actually. I included them because when I studied for this final, I had no idea where to start, but I kind of read through the objectives and I was like, can I answer this question? And if I could, I like kind of moved on, and it actually worked really well. So that was my personal opinion. So what is an amino acid? Do you all remember what an amino acid is? Kind of, hopefully. Yeah. Amino acids are made up of molecules, and they're the building blocks of proteins. So these molecules, hydrogen, nitrogen, carbon, oxygen, make up amino acids, sometimes sulfur. Yeah. They bond with each other, make up proteins. You've got your amino group, which is the NH, and then you've got your carboxyl group, which is the COOH, and then you've got your central carbon, your H, your hydrogen attached to the central carbon, and your side chain. And this is just the smallest unit, which could be an amino acid, right? On their own, the atoms and functional groups that make up amino acids are not amino acids until they're all together. So this is the smallest group of molecules that would be considered an amino acid.
Does anybody have any questions about the structure? You all got it? Cool.
Very cool. What's a protein? Amino acid backbones form bonds with each other called peptide bonds in order to create proteins. Therefore, proteins are just long chains of amino acids folded in different ways. My metaphor for proteins and folding is a little bit specific and weird, so if it doesn't help you, please disregard this. But what I like to tell my friends is that it's kind of like when you crochet, you're starting with like this long string and you're turning it into like a larger item, like a sweater or a scarf or something, and all of these items do different things, obviously, depending on what you wanted them to be, but they all started from like this long chain. So if you're thinking to yourself, how do I make a protein out of this chain of amino acids? It's kind of like when you knit or crochet. Yeah, but obviously the folding is much different. So I wanted to talk about level of structure, but I'm getting ahead of myself a little bit because I think these images are helpful. So you've got your primary structure, which is literally the sequence of amino acids because they're all different, so the sequence matters. And then you've got your secondary protein structure, which is made up of beta sheets or alpha helices, as you can see here. The beta sheets, they look like a piece of paper, and the helices look like spaghetti noodles, I guess. Then you've got tertiary protein structure, which is formed via the R group interactions, which are the individual and unique parts. So all of the R groups have their own unique chemistry, which is what drives the R group interactions. And then you've got quaternary protein structure, which is only for multimeric proteins. So does anyone remember what a multimeric protein is?
Multiple strings of amino acids together?
Yeah, when multiple different separate strings of amino acids form, together.
What exactly is like a beta sheet? Why is it formed to begin with?
Yeah, so a beta sheet is just when the amino acid string kind of looks like that and it goes past itself. Yeah, like that. So the amino acid string is literally folding past itself, and all you need to know is that there are interactions between the backbones of those amino acids that are next to each other. So if this is part of one chain and this is part of the other chain, they're sitting next to each other in space and forming interactions with each other.
So what would be forming the bonds between the two?
The backbones.
Yeah, is that like the hydrogen?
Oh, yeah. Yeah.
The structure.
Yeah.
One more.
What?
Back.
Oh, back. Yeah.
So yeah, it's all the backbone things. So anything but the side chain, these can have interactions with other amino acids, backbones. Those will make covalent bonds.
Yeah.
Or not covalent.
Not
sorry,
covalent.
Non-covalent.
Non-covalent.
bonds, my
Hydrogen
bad.
bonds.
Yeah.
Yeah, they will form hydrogen bonds because you've got the NH and then the O's, and they can interact with each other. Yeah.
For tertiary, what are the interactions, what bonds form?
What? Oh, what bonds form? Yeah. So here it is. Mostly non-covalent, but they can be covalent. And I was, just getting to that. So here we go. Here's this chart that I put together. So your level of structure, how it's held together, and the types of proteins that have this structure, and are they non-covalent or covalent? Can anyone tell me why it's important to know whether or not a bond is covalent or non-covalent? I'm going to call in someone else. Unless someone else doesn't want to participate.
Come on, guys, free candy. There you go.
So like bond strength, I know that in an aqueous environment, the covalent bond's a lot stronger
Yeah.
than the non-covalent.
Right, yeah. But why is bond strength helpful? Why is knowing how strong your bond is?
Protein structure.
Yeah, to determine how stuck together these molecules are in any given situation. Right. Yeah. So, what I like to think about the difference between covalent and non-covalent bonds is that covalent bonds are not breakable or makeable spontaneously. You require an enzyme to form them. So, when you have something covalently bonded, you know it's a molecule that the cell, it's really important that the cell is not spontaneously just degrading or on making this. So when we talked about, for example, phosphorylation, that's a covalent bond, which is done by an enzyme. And that's something you don't just want happening, right? But a non-covalent bond is something that could happen spontaneously in the cell because they're weaker, so they could just happen if the molecules are close enough to each other, which which is good because it allows the cell to do interactions at a lower energy cost, because all of the bond formations cost energy.
If you think about it, most covalent bonds, if not all of them, require an enzyme to be made. So if protein folding was to happen just on its own in a cell, it'll probably mostly form non-covalent bonds. There's a few exceptions in which they're covalent and they don't need an enzyme, but mostly they'll form non-covalent bonds, again, by themselves. Yeah.
For endergonic reactions, it's going to be from small to big. It's creating bigger molecules from smaller ones, and those are most commonly facilitated by enzymes because they have they need energy.
Yeah.
Yep.
But for non-covalent, it's still endergonic, small to big.
Yeah.
It's just a lower level.
Yeah, exactly. Because it's lower level, even though you're trying to create new bonds and you're forming new bonds, because the lower level of energy, because it requires a lower level of energy, you don't need a catalyst for it, which is why it's favorable to have a lot of things be driven by non-covalent interactions.
Okay, awesome. Thank you.
The thing that is forming the covalent bonds for primary structure is RNA, right? Or ribosomes.
Ribosomes.
There you go.
Yeah.
What about disulfide bonds? What structure would that be?
Disulfide bonds are found in tertiary or quaternary structure. They can help to stabilize either. And disulfide bonds being covalent, I'm pretty sure enzymes are needed for those.
And those are between the cysteine?
Cytosine, yeah.
Cytosine?
Yeah.
Oh, my bad.
Wait, not.
I'll get you
Wait,
another one.
cytosine isn't nucleotide based, sorry. Cysteine. Yes, cysteine.
Cysteine sulfide, it's sulfide bonds between cysteine amino.
Yeah. So if you have, So if you have two cysteine amino acids sitting near each other, they can form a disulfide bond with each other. But a cysteine wouldn't form a disulfide bond with itself because you need disulfide, you need two. And cysteines have one each. Yeah.
And those are non-covalent.
Those are covalent.
Covalent.
Yes. Those are probably the only covalent R group interactions we talk really about in depth.
Disulfides?
Yeah. Yeah. Okay, cellular chemistry. We've already talked a lot about this already, which is why I made this slide really short, because I think everyone's pretty familiar with this already. But if you have a polar bond, it's like the electron sharing is very unequal because one of these molecules is going to spend a lot more time with the electrons. One of these molecules is going to have a lot more pull, and the electrons are just going to be with it more, which means it's going to have more of a negative charge. In a nonpolar covalent bond, you would have an equal sharing of electrons due to the similar electronegativity, which means you're not really going to experience differences throughout your molecule, right? If you could see in this one, it pretty much looks the same, which means that it's not going to have strong charge moments. So if you think about magnets, for example, which are also things that kind of have charge, right? If you have a magnet, it's going to interact with other magnets because it's got magnetic fields.
You can think about it like one side is just different than the other, and the difference is how well it can pull electrons toward itself. Like if you have yeah, I guess a magnet is a good
Yeah.
example. You have one side that's different, one side that's not, or one side that's charged, one side that's less charged, or however it is, right? They're different, so they interact differently with other magnets.
Yeah. Okay. Amino acid chemistry. So Laura said she's not going to give you this chart on the exam, right? That's what she said
Yeah.
in her email? Yeah, that's why I was pretty sure. That's why I included it here. You don't need to memorize them, right? Because she said she's not including it, which don't worry about memorizing them. Don't stress about her not including it. I wanted to emphasize the importance of understanding and being able to talk about different types of polarity. So if she said you have a non-polar amino acid being changed to a negatively charged amino acid, what's going to happen?
It won't bond easily?
Well, they'll bond differently, but what's the difference? Does someone want to explain? Sure.
It's hydrophobic and hydrophilic interaction. So polar would be and non-polar would be hydrophobic.
Yeah.
So if it's a change between the two, the folding would change so that the phobic parts would now be on the inside of the protein and it would be away from the aqueous.
Yeah.
Correct.
Yeah. But if you're changing from a non-polar to a polar, what would happen?
It would, instead of folding in, it would fold out.
Yeah, exactly. So just like knowing what she's asking, if she gives you an amino acid, what she's probably going to say is like you're changing this amino acid to that amino acid, and you need to talk about how the size of the molecule matters and how the charge of the molecule matters. So if you change a glycine to a tryptophan, that's going to be a pretty big change, even though they're both nonpolar, because glycine is really small, but tryptophan is huge. Tryptophan is like one of the biggest amino acids. But if you're changing a glycine to an alanine, that change is not that big, right? Because you're just adding a methyl group instead of a hydrogen. So you just need to be able to discuss the level of change you might see. Because obviously you can't say for certainty anything about them because you haven't done the experiment, and that's okay. You just need to be able to use the information about charge and size in order to determine what might happen. Yeah.
Why is the charge important for the amino acids?
Yeah, so charge is important because the charge determines its tertiary structure folding. So, if we all remember, nonpolar molecules don't like polar molecules. So in a protein, if you have a lot of nonpolar molecules, they're going to sit together and hide away from the water. And if you have a lot of charged or polar molecules, they're going to sit on the outside, and they're going to want to interact with the water. So if you change the charge, you're going to change its interactions with the water and with other molecules around it. Because if you've got a polar molecule or a nonpolar molecule on the inside of a protein, and suddenly it's a polar molecule on the inside of a protein, first of all, it's not going to want to interact with the other nonpolar molecules on the inside of that protein, but it's also going to want to get away and interact with water. So it's going to change the structure significantly if you change the charge. Yeah.
So, is there specific amino acids we should recall that have a strong negative charge or a strong positive charge?
You don't necessarily need to know the charges of them. You just need to be able to talk about it if given an amino acid and its charge. I think she would give you the charge, right?
She would
Yeah,
give you the
because she's not giving you the
structure,
table, so she's going to give you the charge. She's going to tell you this amino acid is this charged, or this amino acid is nonpolar, or this amino acid is charged negatively.
So, if an amino acid is not charged and it becomes charged, what happens?
So
Yeah.
if you were given a question where it's let's change this amino acid in a protein from not charged, nonpolar, to let's say positively charged. Yeah. So again, I think you mentioned this earlier. If it's nonpolar, it tends to be folded inwards because it doesn't want to interact with the aqueous environment of the cell. The cytosol is mostly it's water. So if it becomes charged, the protein will want to fold in a way so that new amino acid is kind of positioned towards the outside. That's going to go on and affect how it interacts in space with other proteins. Generally, its structure will be changed, and if its structure is changed, its functions change. That's the biggest thing.
Yeah. You've had your hand up for a while. Sorry. Yeah.
If she asks on the exam, if a molecule is polar, if a molecule is negatively charged, could you treat that the same? Because I don't really know the difference between something. How can something be polar?
Yeah. So the polar molecules, so if you take, for example, maybe serine, it's polar because it's pretty small, right? But it's got that OH group.
I'm gonna back a slide real quick.
Yeah, sure.
Yeah, so like, it's polar, but it doesn't have to be charged. It can have like a tendency to be more negative towards one side of that molecule, or sorry, one side of that side chain. The other side of it could be more positively charged. So it has polar characteristic. But yes, positively charged and negatively charged have the most polar characteristic. It's the most like different.
Yeah, they're more polar.
You got a question? Yeah.
So we will have to know like the size of the amino acid given.
You'll be given an amino acid. Like you might be given, let's say, tryptophan. You'll get the diagram. You'll get its polarity. You'll get everything.
Yeah.
You just need to discuss that. Yes, it's a large amino acid compared to some of the others. So just know generally like this is large. That's small. These are kind of like medium. This is long. Something like that.
Yeah.
Yeah.
Or if she gives it to you and say she didn't give you a diagram, she's almost definitely going to give you a diagram if she asks you for an amino acid. But if she didn't, you could talk about hypothetically, if you said, if this was a small molecule, then a big molecule would cause a problem because it takes up more space. You know what I'm saying? Or you could say, if this molecule was large and it was changed to a small molecule, it would take up less space, and that would also be a problem. Yeah. So you could talk about the hypothesis of it, which would also help you demonstrate your knowledge. Yeah.
The only thing that we probably have to kind of remember is the lysine arginine is positively charged because of the histone relation, right?
Oh, when you're talking about histone modifications. Yeah, you should remember that for histone modifications, but in terms of just amino acids on their own, you didn't need to, for this topic, you didn't need to know that. But yeah, when you get to histone modifications, that is important information. That is a good point.
Topic 10.
Yes. Okay.
All right.
Anyone have any more questions? I know this is a big one because you all know it's coming up. If there's anything I can clarify, please.
Cool.
All right. Oh, yes.
Will negative charge and positive charge be brought up, or?
If you change something from a positive to a negative charge, that would be a pretty big change also, because the positive charges are going to interact with negatively charged things, and negatively charged things are going to interact with positively charged things. And by interact, I mean they're going to attract. But positively charged will repel other positive charges. So if you had two positively charged molecules sitting next to each other, but one of them was suddenly changed, if you had a positive and a negative molecule sitting next to each other, but one of them was suddenly changed to the other charge, they're going to want to repel now. So that is pretty significant. That's a good question.
Can you call that positive-negative interaction, or is there like a word?
Positive-negative interaction,
That's pretty good,
basically,
yeah.
yeah. They would experience technically an attraction force because positives and negatives attracts, and negatives and positives repel. But if you don't remember that specifically, you can just call it an interaction, and you would say that the interaction changes. Yeah. Denaturants. This is a really, really quick one because it's not super talked about in this class, but if you see any of these words in a question, just know she's probably talking about the denaturation of proteins. So pH changes can affect the interactive chemistry or properties of amino acids in a relative sense. So hypothetically, if you change the pH of the environment, your polar charged R groups could become nonpolar, which is bad, because we just determined that changing the polarity of these molecules changes its function, which we don't want, which would cause denaturation. You don't need to know anything about that. You just need to know that changing the pH has an effect. Light adds energy to systems. Increased energy means they're not going to want to sit where they were. They're going to move more. It's probably going to denature your protein. Same with heat. Detergents will physically interfere with and sometimes will even just break your bonds. So just know that if she asks you about like something and she calls it a detergent, it's going to denature your protein. Or if she says she's adding a detergent to denature it, you know she's trying to change the structure.
I would just say one more thing to remember. In terms of pH, does anybody know what pH measures? Acidity, but like acidity of what? Like what is it? What specific ion does it measure in the cell or in anything? Hydrogen, right?
Yeah.
So it measures availability of hydrogen. If there's a lot of hydrogen, it's a low pH, but that also means those hydrogens can go and bind other things. You don't need to know that specifically, but just for your own understanding of how it works, when they go and bind other things, they'll change their polarity a little bit.
It
So
changes
that's
charge.
kind of how it works. It might help you clear things up mentally, but she might ask, how does pH, or like change in pH does what? You'll say it'll affect polarity of the amino acids, which will then result in denaturing of the protein, which
Yeah.
will then result in unfolding, whatever you want to say.
Yeah, and this diagram is just showing you, like, if you have a folded protein, your denatured protein is just going to be unfolded in some way, and then it'll get tagged for ubiquitination, and it'll get degraded.
Which denaturants affect which kind of bonds?
No, you don't need to know that. That's way too much detail, yeah. It's not likely she'll even ask you about denaturants. She just, if you get asked about it, it'll just be like, which one, like, will this denature your protein? And you have to just be like, yes. Yeah. Any other questions? Because I think this is the end of topic 2. Oh, here's a question. Sorry. Never mind. Any questions before we go on to questions for you? Okay. Questions. Say I have a protein made up of 120 amino acids, and I'm trying to find the 100th amino acid. Which terminus is this amino acid closer to? Yeah, C. Why? Yeah, do you know why?
Ranges from N terminus to C terminus.
Yeah, because the N is the beginning, you start counting at N, and you count to C. Perfect. C terminus, yes. Why do we say that ionic bonds are weaker than covalent bonds?
Because they're easier to break apart.
In a cell, yes.
So,
Yes.
what's the main reason why? Do you know? Because technically, ionic bonds, if they were outside of the cell, could be stronger than covalent bonds.
Yeah.
Do you know why inside the cell they're weaker?
Because of the aqueous environment?
Yep.
Yeah. And for those who didn't hear, the partial charges of the water molecule interact with the ionic molecules and make it so that less energy is needed to break the ionic interactions between two charged molecules.
Yep.
Cool. Aqueous cell environment. Wonderful. There you go. This is you.
Yeah. All right, that was topic 2. On to DNA, RNA transcription, RNA processing. Again, unit objectives. Feel free to go through these. Any quick questions before we get into it? Anybody have any pressing questions? Cool. So the big thing you should really hone in on for this topic is the structure of DNA and RNA. It'll help you understand the later things in this topic. So both DNA and RNA are made up of a phosphate group, a sugar, and a nitrogenous base. As we know, RNA has a U instead of a T. That's one of the main determining things. And also, they're polar. So one side of the molecule, or one end of it, does not look like the other. So that's another way you can think of like what the word polar means. They're also polar in the sense that we just talked about in topic 2. They're made up of many charged atoms that cause it to be positive in some locations, negative in some locations, things of that nature. Does everybody understand what I mean when I say 5' end and 3' end? Any questions there? So you guys get that the 5' end, sorry. The 5' end is up here at the 5th numbered sugar, and then the 3' end is here at the 3 numbered sugar. You guys get that? Cool.
Everyone remember the difference between DNA and RNA in structure?
Cool. Yeah, that's the biggest thing you should remember, 5' and 3', and that RNA and DNA differ also in their 2-sugar bond to either OH or H. So the reason it's called deoxyribonucleic acid in DNA is because it's 1 oxygen less, so deoxy, which is, you can see on the bottom there, it doesn't have the O. On the top, the RNA has the O. So you'll also need to know how transcription is done. RNA polymers, so pieces of RNA, are synthesized from 5' to 3'. So if I go back really quick, it's top to bottom in that structure. Because they're made 5 to 3, both DNA and RNA, they're read in the opposite direction. They're read 3 to 5 and made 5 to 3. Any strand you'll see, if you see the top strand is 5, a bunch of letters, 3, the bottom strand will always be 3, a bunch of letters, 5. Does everybody get that? Cool. So a lot of people may ask, what makes RNA polymerase choose one strand over the other to be the template strand. And that's because transcription factors will bind one of the strands of DNA in a particular orientation, which will determine the orientation of which RNA polymerase will bind. There's a bunch of different levels of detail there. Just know that transcription factors cause it to bind at a certain spot on a certain strand. Cool. The bonds between each molecule of DNA or RNA are called phosphodiester bonds. So you probably need to know that name.
Sorry, what did you just say was the bonds between?
They're called
Phosphodiester.
phosphodiester. Yeah.
And what's it between? Those are the ones between phosphates.
So
It's between the 3' OH group of one of the molecules and the 5' phosphate group of the one next DNA.
basically this phosphate group
stack.
up here
Yeah.
at the 5 end will bind to something bound at this 3 end.
Yeah.
And so phosphate ester, it's another structure you don't need to know, but you combine the words, it becomes phosphodiester. Yeah.
Does the 3' of both pentose sugars have a hydroxyl?
It does, yeah.
Yeah, the 3' always has a hydroxyl group. They just didn't show it, which is poor diagramming.
Yeah, that's true.
Another
Right.
slide.
Yeah, that's true. No, I was just seeing if I missed anything.
Oh, yeah.
Phosphodiester bonds, yep. So RNA gets processed in eukaryotes. So they don't get processed in prokaryotes. Can anybody tell me why they don't get processed in prokaryotes?
Because prokaryotes have continuous, can do transcription and translation simultaneously and
Yep.
don't really have a break like eukaryotes.
Yep, correct. And so if eukaryotes did transcription and translation together, they would not need RNA processing. But it's needed in eukaryotes. Why? Because the RNA is unstable in natural form. It needs to have different things added to it so it can be stable and regulated, basically. So the 5' guanine cap is added for stability. Introns, so this happens in eukaryotes, not in prokaryotes, but introns get spliced out. Those are different regions of of RNA that don't lead to amino acids being translated in the ribosome. So those get basically taken out of the RNA. So that, she may ask you this, she may not, but it results in different, it gives variability to those genes if those are spliced differently. Basically, if different chunks are taken out, it results in a different gene.
Yeah, that's an important one, actually. Differential expression and splicing. A lot of people confuse differential expression and splicing because splicing allows for differential expression in some cases, but differential expression is not always done via splicing.
Yep. And then lastly, there's a 3' poly A tail, again required for stability, but also this one's especially important because it's required for export out of the nucleus. So you guys need to remember that. Without the poly A tail, it can't leave the nucleus and then it gets degraded.
Is that the one that prevents like the nucleases from coming and chewing on the end of it like that?
Yep.
Yes.
Correct.
And actually, it also kind of serves as like a timer because depending on how long the poly A tail is, that means that's how long that piece of mRNA transcript will be, which also helps to determine how many times it will be translated. So because the poly A tail is also chewed back, just like the rest of the mRNA, but it prevents the mRNA itself from being degraded immediately. But once it gets to the mRNA, then the mRNA is also degraded.
Basically, if it runs out of poly A, if it runs out of
Yeah.
A, it'll start chewing on the mRNA
Yeah.
and then get degraded.
Right, okay.
It just delays that.
Yeah.
Is it protection from the nucleases or nucleosomes?
Nucleases. You want to give him a piece of candy?
Hmm?
You want to give him a piece of candy?
Okay. Sorry, my throwing is really bad. I'm gonna walk.
All right. Here is a piece of DNA. We're going to say that the top strand is the coding strand. So can anybody tell me which way transcription goes?
From 3 to 5.
Yeah, I meant left to right or right to left.
Oh, the top.
So the top is the coding strand, which means which way does the transcription go? Right to left or left to right?
Right to left.
Right to left is right. So the top strand is the coding strand, which means the bottom strand is called the template strand. And because
Sorry.
because the bottom is the template strand, it gets read 3 to 5, aka left to right, which means it has to be made 5 to 3, which is right to left for the bottom strand. Everybody clear on that? Cool? Cool.
Yay.
So that was topic 3. Any last questions before we move on to topic 4?
You definitely need to know how to look at DNA and kind of understand it and not be scared of, don't be scared of looking at DNA. I had a friend who was like really scared of DNA and I felt so bad for her because that's so valid, because it's a lot to look at. There's a lot of letters, but don't be scared of it, guys. It's like, if you look at it really slowly, like you're like reading a book, you can kind of figure it out if you know what you're looking for. Don't get overwhelmed by all the letters is what I'm saying.
Cool.
Yeah. Topic 4.
All you, man.
Mechanism of translation. Here are your goals. Here are things to think about, to focus on. Show gets to throw the candy so no one gets hit.
I'm actually gonna you one.
Types of mRNA. Darn it. Types of mRNA involved. So here are your 3 types of mRNA that we talk about in class. So you've got mRNA, tRNA, and rRNA. Is everyone good with all of those types of RNA? Yeah, probably. This is what tRNA looks like. It's actually really blurry. I don't know why, but I apologize. This is actually in the notes, so if you want to reference the notes, you can see a clearer version of this. But yeah, so this is a diagram of what tRNA looks like, basically as a drawing of the backbone and nucleotide bases. Like, this is like just a really basic outline of what we like kind of, like if you flattened a tRNA, that's what it would look like. But that's not what it actually looks like in 3 dimensions. Here's its 3-dimensional structure, which is a little scarier. And here's your space film model. I just thought this was a fun diagram to include. This top is where your amino acid will get added, and this bottom part is what base pairs with RNA. Is everyone good with the structure of tRNA? Probably. Yeah. Oh no. Okay. I've included this elongation diagram because initiation is different for prokaryotes and eukaryotes, but I honestly think that's probably not going to get touched upon, so that's why I didn't really talk about it. The only real difference is that, well, does anyone remember the difference? Maybe? Yeah?
In prokaryotes, the tRNA binds to a subunit of 4. The
Yeah.
mRNA,
Yeah.
But in eukaryotes, it's the other way around.
Yeah. Wait. In prokaryotes, the ribosome, no, I think you've got it backwards. Wait, what did you say?
That in prokaryotes, the tRNA binds to the subunit, or like the Schindel Gardner.
Yeah, okay. I think you've got it backwards. Eukaryotes, it binds first to the start codon, to the tRNA. Yeah, the large subunit in eukaryotes binds to the tRNA in eukaryotes. Yes. And then in prokaryotes, first it binds to the mRNA directly. And then the big subunit comes, and then your tRNAs enter. So, E, P, and A sites. Does everyone remember what E, P, and A sites do, what they are? Which direction do they go? A to P to E. Perfect, yeah. So you have them entering at the A site, you have them going through to the P site, and then the next one comes and enters the A site when the second one's in the P site, and then they all move over again, and then another one would come in here at the A site and get tagged along, and here's the exit site, and you can see the exit site, it's no longer connected to its amino acid, that's because it's leaving, and this one's getting attached to there, and your new one's coming in and sitting over here. And yeah, the peptide bond formation happens between the A and the P sites.
Yeah.
Where's it held?
Hmm?
Where's it held?
What do you in the cytosol?
The peptide chain that's being formed, is it held in the P or the A?
Oh, the peptide bond chain. Actually, this diagram shows it as being held in the ribosome, but that's not technically really true, because while the formation of the peptide bond happens in the ribosome, the protein actually gets fed out of the ribosome and then starts folding, like, right away, like,
In the
as it's
cytosol.
being formed. So it folds from the N-terminus to the C-terminus, because the N-terminus leaves the ribosome first and starts folding in the cytosol. Or sometimes in the endoplasmic reticulum, but you don't need to know any of that. Yes?
The amino acids being coded for, is it the codons of the mRNA or the anti-codons?
The amino acids that are being coded for. Okay. Sorry, can you say that one more time?
So you know the table that we're given? And
Yes.
It's like, okay.
It's the
like,
mRNA.
Yeah,
It's the mRNA.
yeah,
It's the
yeah,
mRNA.
it's the mRNA coding table. Yeah. That was a good question. Yes.
Think anticodon, meaning anti, like not. So it'll
Not the
always,
codon,
yeah.
the opposite of the codon.
Right.
Yeah. So the codon is part of the mRNA.
Yep.
Yes. Okay, so understanding frames and mutations. So first, can someone explain what a frame is? If not, I'll explain it. But does anyone know, and can anyone explain it first? Yeah?
Isn't it the set of like the 3 nucleotides after the start codon?
Yes. Kind of. Yes, basically. Yeah. It's really important that you have a very specific understanding of frames, because it's a very complicated thing that actually took a really long time for me to understand too. So yeah, it's set by the start codon.
You have a question?
So, so is the start codon the same as the stop codon in the way that it doesn't actually code for anything, or does okay?
No, the
Does-
start codon codes for methionine.
Okay.
Almost always methionine.
Okay.
Sometimes something else, but usually methionine.
Okay.
Did you have a question? Okay. I saw your hand up, but yeah. Yeah. So the start codon sets the frame, and it's like you set the frame of the first 3, and then the next 3 get read. But if the start codon was moved over, then the same next 3 would not be the same. You know what I mean? It's like moving a slider along like a bar and then just stopping it. So if you had your DNA, and you slide along the bar and then you stop when you find the start codon, and then everything else you can start reading as base pairs of 3, right? Does that kind of make sense to everyone? Yeah. It's kind of a weird topic, but this kind of shows you the different hypothetical frames in this specific strand, and how if you started at different places, you'd get different transcripts entirely. You see that? Yeah, that's why I like this picture. But yeah, these are the 3 different groups. Yeah.
The one's confusing me a little bit because there's no start codon.
Yeah, this is just showing you the different frames that exist. They're not start codon frames. When you actually read it, you have to start with a start codon. But since they're just letters, you can technically set the frame anywhere. Yeah, it's just showing the concept of different differential framing. Yeah. And so point mutations impact your frame setting, which will ultimately impact your transcript a lot. So, the types of mutations that we talk about. Your frame shift mutation, they listed that first. Okay. Frame shift mutation is when you have an insertion or a deletion and your whole frame just moves over 1, right? So as we see in this diagram here, that'll change all of your transcript outcome. So here, if you had it set as every single group at the beginning, you'd have serine, stop, codon, serine, arginine, lysine. But here, if you move it over by 1 nucleotide, 1 singular nucleotide, you get histidine, asparagine, leucine, valine, and arginine. That's really different. Those are entirely different sequences of amino acids, which is why frame shift mutations are really bad. Deletions and insertions, not good. You don't want to be inserting or deleting random nucleotides. You have a silent mutation, which changes 1 letter to another letter, but it'll code for the same exact amino acid. So that actually, does anyone know why silent mutations can still be bad for the cell? Yeah.
A cell mutation can cause a protein not to function the same way.
But if it's coding for the same amino acid, would that change the function? No. But what does it change? Yeah?
Lack of tRNA.
Yeah. Frequency of tRNA. There are certain tRNAs that are more common, so tRNAs with different codons are more common. So if you So hypothetically, UUA could be a lot more frequent than CUA. So if you change it to CUA, you're probably not going to be able to produce this protein at the same rate anymore because of the lack of that one codon. You know what I mean? So that's why even silent mutations can still have a really large effect on your translation. Missense mutation is when you have a mutation that changes it to a different when you have a point mutation that changes it to a different amino acid that's not a stop codon, right? So you're still coding, you haven't stopped it, but you're coding now for a different amino acid, which is what we were talking about earlier. When you change amino acids via point mutations, like in this example, you're changing from a leucine to a valine, you can have problems. And lastly, you have nonsense mutations, which are just premature stop codons. So you've changed this from a UUA to a UAA, which is a stop codon. So it will stop early, which means your protein is going to be incomplete. Yeah? Cool. Question. In the above...
The answer's already given. Oops.
Oops. I don't know. Yeah. Does anyone want to come up and talk about how you might find these start sites? You don't have to come up. You can just talk about it. You can just talk about it. Someone new, maybe?
All right, I'll give you a hint. Oh, yeah, you got it?
If you read 3 to 5, TSC, it'll be 5 to 3, I think AUG.
Yep, that's correct.
So it's 3 to 5, right?
Yeah.
So could you, follow-up question, could you tell me why it's CAT on the top but TAC on the bottom?
Well, up top is, from left to right is 5 to 3, but 3 to 5 is...
Good.
Yeah.
Good.
Sounds like a stupid question, but it's sometimes hard to remember if you're worried about taking a test. So, keeping in mind directionality. And that is topic 3. Oh, well, that just says 3.
Those
Yay,
red
3.
boxes are supposed to come later.
How many red boxes are there, guys? 3!
Alright, sweet.
Yay. Anyone have any questions on topic 4? Yeah.
So for transcription, it's red 5 to 3, made 5, red 3 to 5, made 5 to 3.
Yes.
Translation is red 5 to 3, made and C.
It's red from
5 to 3.
5 to 3?
Yep.
Okay, yeah.
You don't necessarily need to know that. It's red technically from just how it presents, because mRNA is just one. But yes, 5 to 3. Yeah.
Yeah, 5 to 3, and then it N to C, yes. Any other questions? Oh, yes.
Is TAC only for DNA and then AUG is for the RNA, or are they looking interchangeable?
Yeah. So, if you're told you're looking at DNA, you're looking for TAC from 3' to 5'. But if you're told you're looking at mRNA, you'd be looking for UAG. AUG, sorry, yeah. AUG, which is the opposite of TAC. Yeah. What? I think you look for it in the 5' to 3' because that's the direction mRNA is read.
Oh yeah, if I missed anybody with candy, just raise your hand. I'll give you a piece.
They all raise their hand right now.
Yeah, I mean, I'll give you candy anyway.
I know you haven't participated. Okay. Topic 5, transcriptional regulation. Here are your objectives. There's a lot of them. This is the Lac-Operon. I love this subject. We were trying to put together slides, and Sho was like, "Oh man, it's the Lac-Operon. I don't want to do that." And I was like, "Wait, I love the Lac-Operon." But yeah, transcription factors. Everyone knows what transcription factors are, hopefully, probably. Transcription factors are proteins. They bind to DNA by interacting with specific sites in the DNA called binding sites, or non-specific sites. I explained that later, but I wrote specific up there.
Yeah, please on the exam don't say transcription factors are enzymes or transcription factors are DNA. Don't say that at all. Laura will not like that. They are proteins.
Yeah, transcription factors exist in the cell. They're proteins. They're coded for in a different region of the cell. They get transcribed, translated, folded, everything, right? And then they go back into the nucleus via a nuclear export sequence, actually. They get trafficked into the nucleus, and then they go and regulate transcription. So they have their own process of being made first before they can go and do their special job. Here's an example of a repressor protein, right? This pink blob is the protein, and this binding site is the binding site for this pink blob. So this repressor will bind to that binding site and do something with the DNA, either the repressors downregulate, actually. Just think about transcription factors in general. Positive versus negative, right. So everyone remembers the difference between positive and negative transcription factors? Anyone want to tell me about that? Yeah.
Positive upregulates, negative downregulates.
When?
When the cell needs it to. I don't know.
It says so on the slide.
When bound to the
Yeah.
Sorry.
No, you're good. I include when bound to the DNA because it's important, because when they're not bound to the DNA, if a negative repressor is not bound to the DNA, you're actually going to see an upregulation of transcription. Does that make sense to everyone? Yeah. So negative repressors, when you're talking about them, aren't always going to stop repression. So if you're talking about a negative repressor falling off of the DNA, that's going to cause more expression, right? So you have to really think about what the question is asking you. Reading is important. Did you have a question? Okay. You just wanted to say when bound to the DNA. Yes, so positive upregulates, negative downregulates when they're bound, opposite when not bound. General transcription factors bind non-specifically because they regulate many genes, a lot of genes. Each of them regulates a large set of genes. But the regulatory transcription factors bind with a lot of specificity because they're only needed in certain circumstances. Okay. Here's everyone's favorite table. Yeah, that's how a lot of people feel, I think. So, how I like to talk about this table is think about it really methodically, because if you're asked about skin cells, for example, right? If the question is, what genes are expressed in skin cells, right? You would start with the skin cell line, type 3. These are your types of cells, and you would see which transcription factors are being expressed. So each of these transcription factors are physical proteins that could or could not be present in the cell. In skin cells, you have physically present A, B, D, and E, always, unless you're told there's a mutation that causes one of them to not be physically expressed in the cell. Now, all of your skin cells have the same genome, but they're not expressing all of these genomes. So skin cells would express, they don't have both bending proteins, so there's no bending issues. So you would need A, B, and D, because B and D work together, right? Yeah. And this diagram will actually tell you which proteins you need at the same time, right? So you'd need A, D, and B, or A, E, and C. You don't want all of them at the same time. You also don't need all of them at the same time. If you only have one activator protein or transcription factor, that's okay, as long as its specific bending protein is still there. So then in gene 1, gene 1 will be expressed in skin cells. Gene 2 will not be expressed in skin cells because you would need the binding site for transcription factor B in order for it to be expressed in skin cells. Gene 3 has the binding site for A, the binding site for B, and the binding site for D, and it does not have the binding sites for, and it does not have the binding sites for these two, but that's okay because you don't need those two. So now it can express gene 3. And gene 4 has binding sites for B, C, and D, but it doesn't have a binding site for E, but that's okay because all you wanted is B and D and A, so that's okay. So it will only B and D. Yeah, it will only not express gene 2. Yes. I know that was a lot of words, I'm so sorry.
That's okay. Are B and C the ones that interfere with each other and make
Yes.
it through a whole loop?
Yes.
This, yeah, go for it.
If gene 1 has binding site for C and D, so should it not transcribe into cell type 1 because it do two loop at once?
So genes number 1, 2, 3, and 4 don't correspond to cell type 1, 2, 3, and 4. All of these cells have all of these genes. So each liver cells have all of these genes. Spleen cells have all of these genes. Skin cells have all of these genes. And muscle cells have all of these genes. What changes in each of these cells is which of these genes are being expressed. For skin cells, we determined all 1, 3, and 4 are being expressed, but gene 2 is not being expressed. Yeah, you first and then you.
So why is gene 3 and gene 4 expressed in skin if E is not active?
Oh, wait. Shoot. B, E. Yeah, you're right. It's only gene 1 that's being expressed. That's my bad, guys. Yes.
The basis behind this is that eukaryotes have their both regulatory and general transcription factors throughout the like the genome,
Yeah.
or is it just
well,
regulatory?
It,
'Cause
not-
they use the bending protein
Yeah.
so that they can
Yeah.
have the regulatory to
Yeah. I wouldn't say throughout the genome. I would say throughout the cell, they'll both have regulatory and general transcription factors, but the binding sites that are expressed in different genes. The transcription factors that are expressed, the type of transcription factor is different in different cells, but they all have them floating around.
So I guess my question is E and D regulatory or is it general?
Oh, I'm pretty sure they're regulatory, right? Are D and E regulatory or general transcription factors? I think they're regulatory.
they activate RNA polymerase
Yeah.
if it can contact. So
Yeah.
activating
But they're-
wouldn't be.
No, but they're regulatory, not general, because they're binding with specificity.
Oh, yeah.
and they're not
okay.
just generally binding
Yeah, that's
and
true.
helping promote RNA.
Yeah.
They're
Regulatory,
regulatory.
yeah.
Yeah.
Would genes reinforce the expected cells?
No, she definitely caught me on that one. That was a mistake that I said, because skin cells, you would need binding sites for B and E only.
I thought it's B and D.
B and E. Or, wait. Guys, I'm confused. Give me a second. B and D work together, and you have B and D, and you need binding sites for B and D. Yeah. Wait, yeah, no, I was right in the first place. Yeah, I was right the first. Sorry, I'm confusing myself because it's very complicated. Yeah, skin cells would express all genes except gene 2 because B and D, not B and E. You're very right. I'm so sorry.
Why do the liver cells need both C and B, like the transcription factors for those? Because wouldn't that turn it all the way around? Because I know
Yeah.
that those transcription factors bind to the DNA to allow
Yeah.
it to bend.
Yeah.
So if both of them need to be active, wouldn't it just fully
Yeah.
turn it around?
That's a good point. This is getting into why they're all different. Some of them will express different transcription factors in order to prevent them from expressing genes that they don't want to express. Because all of your cells express different genes. So liver cells, because they express both bending proteins, will only express gene 2 and 3 if the other binding sites are appropriate. I didn't check, so don't, But they will only be able to, hypothetically, because in genes 1 and 4, both binding proteins are present in liver cells and can bind, and therefore it will overbend. And so gene 1 and 4 are probably genes that liver cells don't want to be expressing. So maybe it's a protein that helps with, neuron growth. You wouldn't want that in a liver cell. Yeah. Did that help, actually? Because that was a really good question. Yeah. Okay. I'm so sorry for the confusion. Does anyone have any more questions? Yes.
Are both regulatory and general bound by hydrogen bonds?
Yes.
Good question.
They hydrogen bond with the backbone. Yes. So, operons. Prokaryotes have really small genomes in comparison with eukaryotes. Therefore, they have a lot less genetic material, physically. It's like having the entire dictionary or a little pamphlet of words. It's a lot less. So how do they code for everything? Operons. It's like optimization of organization in prokaryotes because they have to. So there are sections of the genome that are controlled by the same promoter and transcriptional factors, and they all work together in order to complete a task in the cell. So all of these structural genes here, these are the coding regions, will allow for transcription in one mRNA transcript and all of these proteins to be expressed at the same time.
Quickly, why does it make sense that certain genes are grouped together under the same promoter? Like, if you think about it.
They're using combination from a certain cellular response.
Exactly. If you need one protein that's coded by one part, one of those structural genes, most likely you'll need other things to help it work.
Mm-hmm.
So if they're all under the same promoter, which means transcription factor will bind and then activate, and then those genes will be transcribed and translated, you're going to need all of them. So that's why they're under the same promoter.
Yeah. It's like how for the lac operon, one of the genes in the lac operon, I think it's LAC-Y, it might be, don't quote me on that. LAC-Y, one of them transports glucose into the cell, which is important if you want to break down glucose in the cell. Lactose. Oh my god, you're so right. Oh my god. Yeah, one of them transport lactose into the cell so that you can break it down. Because if it's not in the cell, you can't break it down. Right? So yeah, that's why you would want those to be grouped together. So the lactoperon is for breaking down lactose, which is a catabolic process. Does anyone remember what catabolic means? Yeah?
It's gaining energy. It starts at low energy low energy reactants. It goes to high energy products.
That would be anabolic.
Yeah, that's
But
anabolic. But you are
you-
gaining energy by taking energy from your reactants and taking it out of them. So basically, when you break down a molecule, it will release energy. And so technically there's energy leaving the molecule's system, which is why it's going from higher to lower energy state. However, you're getting that energy, and the cell gets to use that energy for other things, often ATP synthesis.
Does anybody know the word anabolic in terms of like going to the gym?
It'd be like to build.
Yeah, exactly.
build
So
muscle.
anabolic, people would remember it as building muscle. So catabolic is the opposite of that, breaking down. So anabolic, building up, and in order to build something up, you need to put energy in, hence. And then catabolic is breaking it down, releasing energy.
Yeah. Okay. So we have all of these parts working as a system together to process lactose as a backup sugar source. Does anyone know why lactose is the backup sugar source? Yeah?
It can be broken down into glucose.
Yeah. But why is it a backup? Yeah? Yeah. But why? Yeah, it's easier. It's accessible. Yeah?
Doesn't it like take less energy?
Yeah, because you have to put energy into breaking down lactose, but glucose just comes as it is and enters glycolysis, which is why it's preferable to have glucose. Yeah.
So she talked about like the numbers, like 10, 0, 1, 3. Can you remind me which of those numbers correspond to the chart right there?
Yeah, we could talk about that. So you're never really going to see 0 unless you have a super repressor, right? So if you remember, super repressor refers to a mutant repressor that's always bound,
Yeah.
right? So that might be when you see 0 because there's no leaky expression.
I thought 0 is when there's a mutation in the promoter so that the general transcription factor can never bind.
That also
works.
could work, yeah.
But super promoter,
Super repressor, yeah.
the super repressor has the same affinity as the regular repressor, which we see is level 1 because of the lethal transcription.
Well, a super repressor would never dissociate, whereas a regular repressor will.
But it still has the same affinity as the repressor.
It's
But if it
it'll
doesn't
have
dissociate,
different affinity. If
It's
it's
different affinity.
well, it
if
doesn't
it's-
it doesn't bind with different affinity, because it binds with different affinity if you have a mutation. Yeah.
So if you think about it, a regular repressor will bind and dissociate at a certain rate. If you have a super repressor, its affinity is intrinsically different because it'll bind stronger and never let go.
So I thought that if we say that like this is just a regular repressor, it has 2 domains, one for lactose and one for the DNA. The only difference between the super repressor is that its lactose domain doesn't exist or is unfunctional, so that it still has the same affinity for the DNA.
That is another form of the super repressor.
Okay.
They can come in multiple different forms depending on how the question's phrased. So in that case,
Okay.
if it didn't have the lactose binding site, it's slightly different than maybe, another super repressor that could bind the DNA and never let go.
Okay.
Yep.
You had a mutation that had cap always binding, you can't along with a normal suppressor, would that be level 3 or level 1?
Cap always binding to camp. So the camp is always on, so it's always promoting expression. So if you had that plus a regular repressor, it would increase, yes. It would increase, so it would probably be about 3. I think that is the situation in which it's probably 3, because you're still repressing it most of the time, but when you're not repressing it, you're upregulating it. So there is that period of time where you will see expression. Not a super high amount, which is why it's 3 and not higher, but not like 1 or 0, or close to 0.
Are we required to know what LAC A
No.
codes for? Because I know
No.
that LAC C was the beta gal, and then LAC Y was the permease.
Yeah. Yeah, but you don't need to really know, that. You and then you.
If the DNA sequence is transcribed, is there any way that LAC C, LAC Y, are separated?
Sorry, wait, say that again?
Oh, so you said that the DNA sequence is transcribed so that they are used together.
Yeah.
But is there like any way that they are separated? Like, isn't that gonna create a change of amino acids that?
Are you saying
Oh.
that it? Can you go back to the other slide?
Yeah, operons, all of the sections of operons technically have their own stop and start codon within them. So, excuse me, I'm just gonna put something here. So, if this is like LAC Z, LAC Y, LAC A, at the beginning you have a start codon for LAC Z, and at the end you have a stop codon for LAC Z. That's
You
where
would
it falls off,
flip
the
the
ribosome falls off. And then you have a start codon here for LAC Y, and then you have a stop codon here for LAC Y. And then you have a start codon here for LAC A, then you have a stop codon here for LAC A. So they're all expressed individually, although part of the same mRNA transcript. That's a very good question.
Sorry.
Yeah.
So, kind of going back to what she was talking about, but when I talked to Laura about like this repressor, and she told me it would be 1 because this has the same affinity as a repressor, because it disassociates sometimes. So, how would this repressor be 0? Is what I'm trying to say, because I don't.
Oh,
know.
yeah. I'm pretty sure super repressor results in 0. That's what I remember. But if Laura told you something else, then she's probably, she's definitely correct. But I just remember super repressor being a 0 because if you have a super repressor, as in you have something blocking transcription always, you're not going to be able to transcribe. It doesn't matter, like, if it's not causing leaky expression, it's not going to be transcribed. I guess it depends on how you're defining super repressor then, because if you're defining it as being able to have leaky expression, then it would probably still be around a 1. But if you're defining it as not having leaky expression, then it would be a 0.
What you guys want to remember is like if you think it's either 1 or 0, just explain why you think it's 1.
Yeah.
Because there's always a spectrum with this kind of thing.
Yeah.
And as long as you can explain why you think there's leaky expression based on your understanding of the structure of something, that's fine. That's totally fine.
Also,
Yeah.
could you explain what level 3 would be? Because I know it's like cap's not bound and repressor's not bound, but I'm kind of confused on
Yeah.
that.
Why don't I talk through all of these 4 scenarios and what I would probably estimate the number to be, and then we can have follow-up questions, because I think a lot of your questions are things we might get to soon. Yeah. Okay. So if you have low glucose and you have lactose available, that's the case in which you will want to express the LAC operon, right? So if you have low glucose available, if you have low glucose, cap will be bound and it will upregulate. And if lactose is available, the repressor will fall off and upregulation will continue. That would be about a 10. That's like the most expression you're going to get unless you have a super activator or something like that, right? Yeah. Was there a question? Oh, you're just agreeing with me. I was just like making sure.
Sorry.
So if you have high glucose, right, and you have lactose unavailable, that's going to be like your quote-unquote normal or ideal situation, right? Because the cell wants to be using glucose, doesn't want to be using lactose. If there's no lactose around, it's not coming into the cell really. So you're going to have about like 0.5 of expression is what I personally say, and that's what I wrote down in my notes. Because the repressor will fall off, right? But you're not upregulating, and the repressor is not mutated to be falling off more or falling off less. So it's like very little. I'd say about 0.5 to 1. Anything in that range is probably acceptable as long as you justify it with CAP is not bound, so there's no upregulation. LAC-I is bound, but there is leaky expression. It's just not zero. That's the conclusion of the second line. So this one, not zero, but very low. Although it does say not expressed, it's not zero, which is why that diagram might feel misleading. If you have low glucose and lactose is unavailable, you're probably going to see about like 3, which is higher than 1, but not entirely high, which is the case we just talked about. So your CAP promoter will be present, so it will be upregulating when the repressor falls off naturally, which means that it will happen a lot when the repressor is not around. Because in a situation with no CAP and the repressor also not around, it's not going to really happen a lot anyways because they're not upregulating the transcription. But if you have upregulation, then when the repressor falls off, it will be upregulated and you will see a decent level of expression. And this last scenario, if you have high glucose and lactose is available, then the repressor will fall off, but you also won't have upregulation, right? So if both are available, it's going to default to glucose, which means that you're also going to probably see about 3, maybe a little less because you're not upregulating it, right? But you're also not like downregulating it either because you're not repressing. So it's probably going to be around 3 to 5. Something in that range would probably be acceptable because you're not upregulating. Just knowing that you don't really want to express a lot if nothing's there. Yeah.
CAMP can work without CAP, right? It just won't be upregulated? Or does it need CAP to work?
The CAP protein needs CAMP.
Oh, sorry.
Yeah.
Okay.
Or opposite, the other way around.
Oh, CAMP needs CAP protein. Yeah,
CAMP
yeah.
stands for CAMP activating protein.
Yeah.
So
Okay,
it activates
yeah.
it.
Okay.
Yeah.
Can anybody tell me why, when lactose is bound to the repressor, it can't bind to the DNA? Somebody else? You've already answered a lot. You've already also answered a lot. Anybody else?
Anyone who hasn't
Yeah.
talked yet?
Yep. And
Yeah.
so change in shape is? There you go.
Yeah. Very cool. Okay. So,
Catch.
just as a review, because we just talked about it, no promoter equals what?
No transcription?
Yeah. Are these individual or are
Yeah,
they gonna?
individual,
Okay. No expression, no matter what. Good. No operator.
No lactose?
No operator, no compressor binding, so then it would be constant
Yeah. Pretty constant, but low levels.
for your time.
Yeah. Low, constant, but relatively low levels. Yeah. Too much expression. Well, yeah.
It can't be repressed. If there's no operator, it can't be repressed
Yeah.
even when it's needed.
Which
so it'll
is
keep transcribing.
why it's quote unquote too much, relatively too much.
Relatively
Yes,
too
relatively
much.
too
correct,
much. Super repressor. We talked about super repressors a decent amount already.
Yeah, you can skip. I mean,
Yeah. Super repressors, no transcription regardless of lactose present. And no repressor. I skipped ahead. If anyone wants free candy for that answer you just read.
Lactose is always being cleaved and like separated to glucose into lactose. Correct.
If there's no repressor. But what if lactose wasn't around?
The cell would die.
Well, no, not necessarily. If there was glucose around,
Oh.
it would be okay. But what happens if there's no repressor? Yeah.
There's
You guys.
There's always transcription happening.
Yeah. Expressed regardless of glucose present. If you have a super repressor and a broken operator, will the lac operon be expressed in the presence of lactose? Broken operator.
It's a broken operator, then it's not anyway.
Yep. Same as no operator.
Yeah. Yeah. Oh, there's no answer for that. Haha. So
No,
that's
I mean, yeah,
yeah.
it was discussion.
But yeah, you were right. Yeah. Now we have time for questions, comments, concerns, discussion, anything you guys want.
Could I ask a question about topic 10 or is that not really happening?
We'll let topics 1 through 5 come through, but I'll answer one about 10 if there's nobody else.
Yeah. We also have an extra half hour. We're probably
Yeah.
going to stop the Zoom now, but if anyone wants